In this week's lab, we are going to be setting up several experiments that are going to highlight different barriers to seed germination, particularly how we can overcome dormancy. The first one is going to be a temperature experiment that we're going to do with both cucumbers and celery. We're going to put 30 seeds of each kind at room temperature and then also in the refrigerator. And we're going to see what the effect temperature has on different kinds of seeds. One of the things that's important to note about these two different plants is that usually cucumbers grow in the summertime and usually celery grows in the wintertime. So that might give you some indication of which seed type would prefer to be at which treatment. And the reason why a seed might have this barrier in general is just so that it doesn't germinate at the wrong time of year. The next experiment we're going to do has to do with water availability, and to do this we're going to set up an experiment with 3 different soil media types, and we're going to use Persian carpet zinnias to see if there is an effect of moisture availability on the ability for these seeds to germinate. So we're going to use a mix that's vermiculite all on one side. We'll use a 2-to-1 vermiculite to potting soil in the middle, and then just potting soil on the end. The idea being that we're creating a moisture gradient by including these 3 soil types within one nursery flat. We would expect that the potting soil would be the highest moisture retaining and the vermiculite the lowest moisture retaining, and so that's we would probably, well, I guess it depends on whether these seeds germinate better after they get wet and dry out or after they're just saturated for a long period of time. So we'll see how that turns out. Next, we're going to set up 2 different experiments to do treatments in the light versus the dark. The first, we're going to set up a whole flat that has basil on one side and cosmos on the other. So we'll set up 2 of those flats. One of them we will put in a mylar bag and keep in the dark, and the other one we will keep in the light. So the idea being for this experiment that some seeds are photosensitive, they require light to germinate. Some seeds aren't photosensitive, so they don't require light to germinate. And it's important to know which is which so that whenever you're starting your seeds, you know whether to either sow them on the surface or to bury them. The second experiment that we're going to do in light versus dark is going to be with mung beans, just like we did last week, but this time we're just going to sprout them in water, and we're going to sprout one set of beans in a clear cup that we sit in the window, and we're going to set another set of these beans in an opaque cup or a cup that's not clear, and we're going to put it in a cupboard in the dark. They're going to stay at approximately the same temperature, but the idea here is we're going to see what effect light has on germination. I've heard that if you want to eat mung beans as sprouts, that they taste better if you grow them in the dark. So this is going to be kind of a fun little experiment with a taste test at the end of it. And then lastly, some seeds require something that's called scarification, where you're actually physically damaging the seed coat of the plant to get it to germinate. This is really true of seeds that require being digested before they will germinate. So it's associated with like an edible fruit, or kind of like mechanical weathering and action, so being ground a bit by soil particles and stuff. And to do this, we're going to use cilantro seeds, which are also called coriander, depending on whether you're using it for the leaf or the seed as a spice. And we're going to do 2 different things to these seeds. First, we're going to soak them in coffee. The coffee soak is to simulate being digested, and so we're using like an acidic solution that's a little bit warm, and we're soaking them for, I think it's probably half an hour, 45 minutes, to see what that does to the seed coat, if it weakens it, and if it allows the seed to germinate better. The second thing that we're going to do is we're going to rough up the seed coat a little bit with some sandpaper, like to simulate mechanical action as if it were grinding against rocks. And then we're going to do no treatment and just see which of these 3 treatments or no treatment, has an effect on the rate of seed germination. And when we do this, we're going to plant 2 seeds for each cell, and we're going to use the same kind of cell flat layout that I just showed you in the basil and cosmos.